Hey everybody, and I don't know why I'm saying that like if this is a new video. I literally just ended the other one. Um, but the next thing that we're going to talk about is nervous tissue, specifically neurons. So we're going to talk about neurons, their characteristics, and we'll talk about their anatomy as well. So neurons are the same thing as nerve cells. So neurons, nerve cells mean the same thing. Just like with muscle cells and a muscle fiber mean the same thing. We'll actually talk about what a nerve fiber means a little bit later. But neurons are nerve cells. So neurons or nerve cells, these are cells that are specialized to transmit messages. They send electrical signals from one cell to another. Okay, so nerve impulses or action potentials, which we already know all about. These neurons, they are excitable. Okay, so these are excitable nerve cells that are specialized to transmit messages. So by excitable, what we mean is that they are both irritable and they are conductive. So in order for a neuron to function the way it should, it needs to be irritable. Okay, it needs to have a certain irritability. And what that means is it has the ability to respond to a stimulus. Okay, so in order for a neuron to carry out its job, it needs to be able to respond to information given to it. So in order for the information that's gathered at a receptor to travel and to move to the central nervous system, all the neurons in the pathway to the central nervous system have to be irritable. They have to be able to respond to that stimulus in order to propagate or send that action potential from the one cell to the next. So not only do they have to be able to respond, but they actually have to transmit that message also. It doesn't really matter if they can get excited and be aware of that stimulus if they can't send that message along. So not only do they have to be able to respond to the message and be aware of that message being sent, but they also have to be able to conduct that message or transmit that message through the length of the cell and then over to the next cell. Okay, so they have to be able to transmit an impulse to effectors or another neuron. Other special characteristics is what we talked about before. Neurons do not carry the ability to divide. We call that amitotic. So neurons are amitotic, meaning that they cannot undergo mitosis. They cannot divide. So these cells have extreme longevity, so they can last for a lifetime. So if you live over 100 years, your neurons are over 100 years, okay? Again, make sure you know that word amitotic. That means without mitosis. So these cells cannot divide, so they live for a very long time. Oh, amitotic is right there, the second word, but make sure you know that word, okay? And they also have a very high metabolic rate. So they require a continuous and uninterrupted, abundant supply of oxygen and glucose. They cannot survive more than a few minutes without oxygen and glucose because the activities within the brain are so energy intensive, just like our muscles, just like we got through talking about. So we need those astrocytes that we just talked about to continually bring oxygen and glucose to the neurons so that they can make the ATP to carry out their functions. Because if not, then if our brain doesn't function, we are dead, okay? So they have a very high metabolic rate, so they need a very, very high and continuous, uninterrupted supply of oxygen and nutrients. And one other thing to know, you can see this picture down here, know that, and we'll talk about the anatomy, you should know a little bit about the anatomy, but we'll talk about it more, is that an impulse travels this way, from the cell body down the axon to the axon terminals. Well, actually, more technically, from the dendrites to the cell body, down the axon to the axon terminals. But we'll talk more about all of that anatomy here in a second. Okay. So these are all the different organelles and stuff that you should know for a neuron. I'm going to name them here, but we're going to talk about them in the next slide. But you have your nucleus and your usual organelles. we're talking about the cell body first. So we're going to talk about the cell body of the neuron first. But the cell body is going to contain the nucleus and other usual organelles. It is the metabolic center of the cell. So this is where everything is taking place. It does have what's called chromatophilic substance. We used to call these Nissl bodies. And again, these names, a lot of these names are named for like scientists that discovered them, but we're actually moving away from those types of names and giving them more scientific names. So Nissl bodies are now being called chromatophilic substance. So what this chromatophilic substance is are clusters of rough ER and ribosomes, and I'll show you that in the next slide here. What they do is they produce membrane and proteins as well, just like the rough ER and ribosomes of our usual cells. Then we also have a Golgi apparatus, which is well developed, okay, and forms an arc or a complete circle around the nucleus. You won't see that in the image in your textbook, but I am going to show you that on the model that we have in the lab. Also mitochondria, we'll look at neurofibrils. I'll show you that as well. And then neurons tend to have a large nucleolus, so we'll talk about why that is here in a second as well. So those different things that were just listed on that previous slide, you have your nucleus and nucleolus, and I'll show you right here, nucleus here, your nucleolus here. You have your chromatophilic substance, that's all this kind of blue system here, that's your rough ER, but here and the whole system of it is called chromatophilic substance. That's that. Golgi apparatus you cannot see on this model. I'm going to show, or on this image, I'm going to show you on the model in the lab. In the next slide, mitochondria, you should be able to see those. Those are here, these little orange bean looking things. Those are your mitochondria. And then neurofibrils, you can't see those on here, but you would see them all throughout the cell body. It's going to be providing its structure, and you'll see some that are coming down here as well. I'm going to show you that in the next model. But neurofibrils are just like the cytoskeleton of our regular cells, and they help to keep the structure of the cell. But then you're also going to see some neurofibrils that go all the way along down the axon. And the reason for that is one thing that I did mention is that, you know, neurons have a large nucleolus. Now, it might not be that apparent in this picture, but neurons have a large nucleolus. And the reason why they have a large nucleolus is because they rely heavily on protein synthesis. Because as you know, ribosomes are made in the nucleolus, and ribosomes make protein. So if we have a very high protein synthesis demand and we need a lot of ribosomes, we're going to have a large nucleolus to make all those ribosomes. So again, neurons have a large nucleolus, or nucleoli, if we're talking about plural, but it has a large nucleolus. And that's because protein synthesis is important in the brain or in neurons. And the reason for that is because of the synthesis or production of neurotransmitters. So neurotransmitters will be produced here in the cell body, and then they'll be transported via vesicles down to the axon terminals. And your synaptic vesicles are going to be found in the axon terminals, which release whatever neurotransmitter is required. So you'll have, we talked about neurofibrils. Neurofibrils are like the cytoskeleton of the neuron and keep its structure, but you're also going to find neurofibrils that extend along the axon in order to transport those vesicles of neurotransmitters from the cell body all the way down to the axon terminals. So that's that. And I'm going to show you all those same structures on the model on the next page. So here's this is a motor neuron. So make sure you know that. How you should know that it's a motor neuron is the fact that there's a muscle right there. So a motor neuron is a neuron that attaches to a muscle that allows for motor output. So there's a muscle there, so you should know that this is a motor neuron. So the different structures that you should know on here, and you'll get that in lab as well, but we might as well go over here as well. So you have your nucleus, which is the white portion. It's everything inside the white portion, but that's your nuclear envelope. Your large nucleolus is right there. Those Nissl bodies are these red circles. So these are those Nissl bodies that we talked about, which is your chromatophilic substance, which acts as your rough ER. You can also see neurofibrils running through. So these are these strands that you see all through here. I'm going to erase my drawing so you can see them a little bit better. And then you can see some of those neurofibrils coming down here. You can't see the end, but those are all coming down here because, again, you need to have some long neurofibrils to send neurotransmitters down to the axon terminals. The Golgi apparatus, so like I said, it's not on the other image, but it's on here, and the Golgi apparatus does form an arc or a complete circle around the nucleus. So this red structure here around the nucleus, that is the Golgi body. Mitochondria you cannot see on this model, so that's okay. But you can see these other things as well. So the gray rod in the middle here, that's your axon. And then since this is a motor neuron and since it's out here next to a muscle, you know that it's not in the brain and spinal cord. So you know that this has to be a peripheral nervous system cell here. And since it's part of the peripheral nervous system, we have this cell right here, this orange cell, making this myelin sheath here in yellow. So if this is a peripheral nervous system neuron, what type of cell is making that myelin sheath? We just talked about it. So that orange cell there should be a Schwann cell, okay, because this is a motor neuron, which means that it's part of the peripheral nervous system, which means that if we have myelin sheath on this axon, it has to be made by a Schwann cell. The myelin sheath that's made in the central nervous system is by something called an oligodendrocyte. So I think that's it for those substances or those structures on this neuron. Okay, so another major region of neurons are processes. So these are fibers that extend from the cell body, and we'll talk about different processes. The first one we're going to talk about are dendrites. Dendrites are the highly branched extensions that come off of the cell body. And anytime we have something that's very folded or very branched, you should know that it's that way in order to increase surface area. So these dendrites are highly branched to increase surface area so it can make more connections with more cells at one time. Okay, so hundreds of highly branched sections that increase surface area to accommodate maximum number of synaptic contacts. Okay, so it can communicate with multiple neurons at one time, and I'll show you what that looks like. So what dendrites do is that they receive signals and conduct impulses toward the cell body. So I'm going to show you an image on the next slide here. So the green ones are dendrites. So what they're going to do is they're going to take incoming information, and actually I'm going to do it in white since this background is black. Here we go. So information is going to come in through the dendrites here, and then it's going to take that information and send it to the cell body. Okay, so dendrites receive signals and conduct and send that information toward the cell body. That is what dendrites do. Those are those extensions there. And then actually, I want to show you something else that's here too. This one here. So like I said, dendrites are highly branched in order to increase surface area. And like your lecture note says, it's in order to carry out maximum synaptic contacts. So this is very simple, a very simple drawing. So here is a nerve cell body, and you can see that it's communicating with three, oh, that's white, I don't want it to be white. There's three, connecting with three separate neurons. Now, this is a very simplified version, so it's only showing 3, but neurons have the ability to make many, many different connections. Again, this is just simplified because it's in a textbook, but there could be tens of neurons connecting to this particular neuron at one time. So it's not only just, because a lot of times when you go over these things in a lecture or something or in a textbook, you'll see like a straight line of neuron to neuron to neuron to neuron. It's not like that. That's a simplified version. These dendrites are highly branched like this because they are made to make multiple connections. So again, this is a simplified version, but there could be many more neurons communicating with this one particular neuron in the middle. So those are dendrites. Then another major process of the neuron are axons. So axons are those long extensions coming from the cell body. So axons are a single process that conducts impulses or action potentials away from the cell body. So if we go here, here's a cell body here. The axon is going to take that information away from the cell body in that direction, down the axon. They're also called a nerve fiber. So in this case, I know before a muscle fiber was like a whole muscle cell, but in this case, a nerve fiber is only talking about the axon. Okay, so a nerve fiber is referencing just the axon. So if we talk about nerve fibers, we're talking about nerve axons. Okay, so a nerve axon can range from less than 1 millimeter, and that's going to be a lot of like your cranial nerves. Those don't need to be very long in some cases, to over 1 meter in length. So we actually have some nerves that go from the spinal cord down the back of your leg all the way to your tippy toes. Okay, so we do have some neurons that are extremely long, while others are very, very short. So they have a large range of lengths. Then you have an area called the axon hillock. The axon hillock is a cone-shaped area at the base of the cell body, and it serves as an action potential trigger zone. So it's that area that's going to determine if we've had enough local depolarizations to reach threshold and send that action potential down the axon. So I'll show you that structure as well. That region area. The axon hillock is this area right here. And actually I'll draw it because it's like a cone, so I'll draw it in that way. But it's this area here. That is the axon hillock. This portion is going to be where the action potential is triggered. So if we reach threshold, this area is going to determine that and then send that action potential down the axon. So that's the axon hillock. That is our action potential trigger zone. And then the axons end at those axon terminals, and all of this should be review for you. They contain synaptic vesicles with neurotransmitters, many different kinds of neurotransmitters. So we have, first of all, we have dopamine, because even though we've only talked about acetylcholine release so far, there are over 50 different types of neurotransmitters. So there's not just acetylcholine, there's much more. We have dopamine. Dopamine is involved in movement, attention, learning, emotion as well. You have high levels of dopamine in reward situations. If you have too much dopamine, you may have a more addictive personality. If you have not enough dopamine, that's when you have, well, in some cases, when you have very low levels of dopamine, are people who experience Parkinson's disease, where they do have trouble controlling their movements and things like that. Then you have serotonin. Serotonin is one of our happy neurotransmitters that keeps us in good spirits and good mood. So they're involved in happy thoughts and rewards, also in learning as well, and memory. Then you have GABA. So GABA is a neurotransmitter that helps to reduce neuron activity. So in times they need to be calm, or in times of rest is when GABA will be released. And then we have histamine, which is involved in allergic reactions. And we have also acetylcholine, which we've already talked about. But like I said, there are 50 or more neurotransmitters, and you're not going to be responsible for them all. Just know the ones that we talked about. Then we have our synaptic cleft and our synapse, but we're actually going to talk about that. It's review, but we're going to talk about that again in a couple of slides here. Okay, so actually, did I bring up yet? No, we're not there yet. Okay, so this is just like your synapse. So a synapse is, you know, the connection between two cells. It's a way to transmit a message from one cell to another. And then the synaptic cleft is the actual physical space in between. But we're going to come back to this here in a second. So another thing about neural anatomy is something we have already brought up, and that is the myelin sheath. The myelin sheath is white in appearance and has a high lipid concentration. This is a fatty, insulated covering that helps to, that helps to protect the axon and helps to transmit signals faster. It is formed by the oligodendrocytes, if we're talking about the central nervous system, and Schwann cells in the peripheral nervous system. So make sure you know that there are two types of cells that make this myelin sheath, and they're going to be different when talking about the central nervous system versus peripheral nervous system. So make sure you get that fact straight in your head. One other thing associated or structure associated with this myelin sheath is something called the neurolemma. The neurolemma is only going to be found in peripheral nervous system myelin sheaths. But what happens is, so you can see this Schwann cell wrapping around the nucleus, and it's actually going to continue to wrap around and create this myelin sheath to create this protective covering. And it's going to do it so much so that it's going to push out the Schwann cell and its cytoplasm out to the end. So that last layer where you can see the nucleus in this kind of like a layer of cytoplasm here, that's actually your neurolemma. Okay, so that is the neurolemma. Do you really need to know the neurolemma? I don't think I asked you about that in the exam, but it's the very, very outside of the myelin sheath. I wouldn't worry about that fact a little too much. You can see myelin sheath on here, like I showed you before. Here is the myelin sheath in yellow here. And we know that this is a Schwann cell because here is a muscle. So this must be a peripheral nervous system nerve. Okay, I'm actually going to close my door because some people just showed up and I know they're going to be loud when they get here. Okay, so let me see where I saw myself. Okay. I closed that door for a reason. Can you please get out? I'm literally in the middle of recording. Okay, so that was my daughter. Okay, so myelin sheath. So again, this is the myelin sheath in yellow. We know that this is a Schwann cell because this is a muscle here. So that means this is a motor neuron, which is going to be a part of the peripheral nervous system. Okay, so one other thing about the myelin sheath, not only does it protect the axon, it electrically insulates it. So what that means is it's actually going to very dramatically increase the rate at which an impulse can travel. So when a neuron or an axon, actually, I should say an axon, when an axon is myelinated, a impulse can travel at 100 meters per second. When it's unmyelinated, it can only travel 1 meter per second. So I'm going to compare this to like a football field. So we're not talking about meters, we're talking about yards, but let's talk about a football field that's 100 yards. So if a axon is myelinated, it can travel from goal line to goal line in a second, okay? In that same amount of time, that second that it takes a myelinated axon to get from goal line to goal line, an unmyelinated axon would only go to the 1 yard line. So a huge difference in distance that could be traveled in a second when you're myelinated versus unmyelinated. It's literally 100 times faster if you're myelinated. well, if the axon is myelinated. One other thing that you should know about neurons is that they're not neurons, but axons. When axons are myelinated, they are not myelinated all the way across continuously. There's actually gaps in this myelin sheath. So you can see here's a gap here, here's a gap here, here's a gap here. Those gaps, we call them the nodes of Ranvier. And the reason for that is when impulses travel along an axon that has been myelinated with this myelin sheath, it doesn't just travel through. What it actually does, and this is a reason why they're able to travel so fast, is it actually jumps from the impulse, jumps from gap to gap, from node of Ranvier to node of Ranvier. And this type of jumping, or this conduction that kind of jumps from one gap to another, we actually call that saltatory. So saltatory conduction. Saltatory conduction. You don't really need to know that word. That's just what it's called. But that just means that these impulses are actually jumping from node to node. And that's part of the reason why it's able to travel or send these impulses so fast. Okay, one clinical aspect is multiple sclerosis. So multiple sclerosis. Oh my God, the puppy's crying. I'm so sorry for these interruptions.
Why did you go here?
Okay. Hopefully that gave you a second to maybe write down the rest of what you're going to write down. Again, sorry for the interruptions, but multiple sclerosis, or MS, this is an autoimmune disease that affects the myelin sheath of the central nervous system, and usually it affects young adults. And so what happens with MS is that it is gradually, the myelin sheath is gradually destroyed and reduced to non-functional hardened lesions called scleroses. So you can see in this cartoon image over here, you have this scarred myelin where it becomes sclerosed and hardened because of the damage of that myelin sheath due to multiple sclerosis. Basically, your immune system is attacking its own myelin sheath. So the loss of myelin as a result of this immune system attack on the myelin proteins causes such a slowdown in impulse travel that impulse conduction eventually ceases. So very dangerous disease because, like I said, the myelin sheath allows a impulse to travel 100 times faster than it would without. So you can imagine that all of these very important functions that rely on these impulses being sent at 100 meters per second, if they're no longer getting that speed and these functions are no longer being able to be carried out at this speed, sometimes it's hard for these neurons to catch up, and you'll lose that sending of impulses once it gets to be, so bad. Most people with multiple sclerosis have a disease course called relapsing remitting. So relapsing remitting. So a lot of people have this relapsing remitting course of this disease, which means that they'll have times where their neurons will actually adapt to this damage, and they'll be able to send signals as they should. But then something can happen, like you can get sick with the flu or have something stressful happen that triggers a flare up, and then you have these impulses being sent slower again. So you'll have these people who feel like they're getting better, and then they'll have a relapse, and things will get worse. And then they'll recover for a little bit, and then they'll relapse again. So most people with MS have this relapse remitting cycle of this disease, which is very frustrating and very sad. Because this myelin sheath is being affected in the central nervous system, a lot of very vital functions, very necessary functions are being affected. So symptoms include things like visual disturbances, which can include blindness or problems controlling your muscles. So you'll have like weakness or clumsiness, Also something called a jagged gait, where people cannot walk correctly. Sometimes because these signals are being sent so slow that sometimes, you can go to, you'll want to start walking, but your nervous system hasn't caught up to that desire to walk, and you can kind of give because your muscles weren't ready to contract yet because these signals are being sent so slow. It can ultimately lead to paralysis, though, after years and years of damage, and your body just no longer has the ability to adapt. It can also lead to things like speech disturbances because your muscles of your mouth can't move as fast as you'd like them to, or urinary incontinence. So both your urinary tract and your digestive tract, the ends of these things, so where you urinate and defecate, are usually, or they are, not usually, they are controlled by two different sphincters. So you have an involuntary sphincter that's controlled by your autonomic nervous system, and then you have a voluntary or skeletal sphincter that you can control. So when you potty train, you are trained to control those voluntary sphincters, the one that you can control. So if you are experiencing multiple sclerosis and you get the urge to go to the bathroom and your involuntary sphincter is relaxed so that you can do so, but you can't react fast enough to hold in that excretion, you may have urinary incontinence. So you may, wet yourself or defecate yourself if you cannot control those muscles fast enough to hold it. So very, very sad disorder, very, very disruptive to people's lives. And this all is due to the destruction of these myelin sheaths that don't allow that same impulse rate to carry on. Let's see if there was more in here. Yeah, so there's a little bit more in this section. Okay. So a little bit more about neuron anatomy is that we have these clusters of cell bodies that occur in different parts of the nervous system. If we have clusters of cell bodies and they're found in the central nervous system, we call those nuclei. So we'll actually get into that in the central nervous system. We'll talk about nuclei that you can find in gray matter of the brain. But if we have a collection of cell bodies, neuron cell bodies, those are called nuclei if we're talking about the central nervous system. If we have a collection of cell bodies outside of the central nervous system, we call those ganglion. One specific kind of ganglion or one specific ganglion we're going to talk about in the peripheral nervous system is the dorsal root ganglion. We won't talk about it now, but we are going to get into that. So you can see that here. Here is the spinal cord. So this is part of the central nervous system. Anything outside of that is not part of the central nervous system. So right here, this area right here, this is a collection of cell bodies. You only see one cell body because, again, it's for simplicity's sake. But here you have, at this part of the spinal cord, and this is only at one part of the spinal cord. This is going to be happening all up and down the spinal cord. But in this region, you have a collection of cell bodies, and that's called a ganglion because we're outside of the spinal cord. So we're outside of the central nervous system. This is now the peripheral nervous system. And so we call this a ganglion. If the central, or if the collection of cell bodies happens somewhere in here, we call that a nuclei. But again, we're going to talk about nuclei more when we get into the central nervous system. So here's a ganglion. That would be a collection of cell bodies there. Now we'll talk about nerve fibers. So remember that when we talk about nerve fibers, these mean nerve axons. So in the central nervous system, when we have bundles of these axons or bundles of nerve fibers, we call them tracts. Okay, so in the central nervous system, bundles of axons are called tracts. Okay, if those collections of axons are myelinated, those tracts are called white matter. If that collection of fibers is unmyelinated, we call that gray matter. Okay, So we have tracts, which are bundles of nerve fibers. If those nerve fibers end up being or are myelinated, we call that white matter. If that collection of axons is unmyelinated, we call that gray matter. I don't know if you can hear it, but I keep hearing Discord ding in the background. So hopefully you guys can wait until I finish answering this question or finish this recording. Okay, so this is just showing you white versus gray matter in the brain as well, because you can see it in the spinal cord. In the spinal cord, the gray matter is on the inside, so this is going to be all unmyelinated nerve fibers, and then here's the white matter on the outside. This is all going to be myelinated nerve fibers. And then in the brain, it's actually flipped. In the brain, the gray matter or the unmyelinated nerve fibers are going to be at the outer edge, while the white matter or the myelinated axons are going to be on the inside. So gray matter on the inside for the spinal cord, but gray matter on the outside for the brain. However, so that's for the most part. For the most part in the brain, you have gray on the outside and white on the inside, but there are small pockets of gray matter on the inside of the brain, but we'll talk about that in the central nervous system. Let me make sure there isn't anything that I wanted to mention. Okay. And then in the peripheral nervous system, when we have bundles of whole neurons, we call those nerves. So if you are talking about a single nerve, a nerve is going to be a bundle of lots of different neurons, and they actually have a very structured organization to them. So this is what a nerve looks like. A nerve is arranged much like a muscle. So you have like single nerve fibers. Oh, I was trying to move something else out of the way. We have single nerve fibers or axons here that are being bundled into fascicles. And then you have the entire nerve. So it's just not just like, but it's very similar to the organization of a muscle. And you have the connective tissue sheets on them as well. So you have endoneurium around a single axon, perineurium around fascicles of these axons, and then you have epineurium around the entire nerve. So again, this organization is very similar to muscle organization. However, we're going to talk about this in the, I think in the next lecture, I think in the central nervous system lecture, we'll go more into the structure of a nerve. And actually, I'm going to stop there. The next video will be on classification of nerves.